This conference will now be recorded.
A very good morning to one and all present here. I'm Ms. Neha Mahajan, Executive Organizer. On behalf of BioSetup Lifesciences, take this honor to welcome our Founder Coordinator, Argadeep Das sir, our Event Coordinator, Dr. Kavita Pal, Dr. Ritu Chandal, Dr. Arpana Sharma, and today's eminent guest speaker, Dr. Renu Agrawal. Faculty members, organizers, and all my dear student participants, I welcome you all for the national webinar on the topic Good Bacteria for Gut Health, organized by BioSetup Life Sciences. I hope you all are safe and good at your end. Before starting the session, a humble request to all of you. Please do not share your audio, video, or screen sharing in order to avoid any kind of disturbance. If you have any doubts during the session, please use the chat box. Filling of the feedback form is mandatory for your attendance and certificate. Now let me tell you about BioSetup Life Science. BioSetup Life Science is a hub of scientific research and learning approved by ISO 9001-2015 certified organization and also MSME registered under Government of India. We have multidisciplinary team of eminent and experienced faculty members, research associates that provide support to aspiring students in life science. We have successfully organized many webinars, workshops, internship programs, e-training programs, certification courses, career counseling, and competitive exam preparation. That is for CSIR, IIT-JAM, GAT-B, IISc, and DBT. We are continuously working for career upgradation of the students. As we all are aware, we have been facing difficult times during the ongoing pandemic since 2 years, but we believe in not stopping and giving quality education to the budding minds through online platform. You must have also heard that empty rakes makes more noise, so it's important to keep filling the bucket of knowledge to the source available. Keeping this in mind, now I would like to introduce our event coordinator, Dr. Kavita Paul and Dr. Ritu Chandal. Dr. Ritu Chandal, she is MBBS MD in biochemistry as well as a very well-renowned speaker and tutor. Ma'am is currently working in Bridge Candy Hospital Trust, Mumbai, Maharashtra. She has also published multiple articles in various national and international journals and academic books. Now let me introduce Dr. Kavita Pal. She is a scientist at Advanced Center of Treatment Research for Cancer, Tata Memorial Center, Mumbai. She has vast experience in field of nanodrug delivery, cancer biology, and clinical studies. She has done her graduation, post-graduation, and PhD from Pune University, India. She has been 10 years of teaching and research experience. Her areas of expertise are nanomedicine, drug delivery system, preclinical, clinical studies. She has 2 patents and 15 publications. She has also completed her MBA in project management. Apart from research work, her interest lies in medical writing as well. Now I would request our Dr. Kavita Pal to introduce our today's eminent speaker, Dr. Renu Agrawal. Over to you, ma'am.
Thank you, Sneha. Hello, a very good morning, ma'am, and a warm welcome to you. Hello to all participants. It is my immense pleasure to introduce our today's speaker, Dr. Renu Agrawal. Renu Agrawal, ma'am, is the ex-chief scientist at CSIR-CFTRI, Mysuru, and a rural program coordinator for outreach activities at CFTRI. Ma'am has over 20 patents, 65 publications, and has won many best publication awards, presented over 160 presentations and more than 650 talks. She has guided many MSc, PhD, and MPhil and PhD students. Ma'am has been nominated as a leader of DST Government of India to represent our country as a team leader of scientific delegations for Asia Meet at Indonesia. Her expertise and experience is in bioinformatics and probiotics. Her research involves lactic acid bacteria, yeast, and bacillus as probiotics, inhibiting harmful diseases and reduce use of antibiotics. It is very difficult to include all the information about Dr. Renu Agrawal in such a short time. Still, I am trying my best. Please excuse me, ma'am, if I miss anything. Dr. Renu Agrawal has been awarded many awards. To name a few, She is scientific fellow. She has won scientific fellow awards such as FAMI, FNASB, FNABS, FASB, and FISBT. She has also won a prestigious award, Karmavir Chakra by Rex UN, Best Women Scientist Award by NABS, Women Achievers from International Guild of Women Achievers, Reflection Excellence Award. Ma'am has been also passionate about helping farmers and especially women farmers, and her contributions resulted in Kisan Ratna Puraskar from JCI International, Abdul Kalam Lifetime Achievement Award, Nari Shakti Award from the Ministry of Women and Child Development, Best Women Scientist Award by NABS. Many more are there in the list. She is editor of 6 and reviewer in 15 research journals and has authored many books and written many book chapters. She is also a board member in many international and national committees. Her programs and interviews are featured on Doordarshan and All India Radio. She plays a very active role in upliftment of rural children and women. Apart from all, she is also a family counselor at Police Helpline Mysore for last 20 years. Ma'am, how do you manage all these together? I am really very inspired and motivated by you and your achievements. Even after retirement, ma'am is active in providing her valuable suggestions as as project advisor, science communicator, reviewer, and visiting professor. She is serving the society. She is a philanthropist and has been running many fellowships. I feel lucky that I got the chance to introduce you, ma'am, and I get motivated by your talk. And you are, I would rather say you are really a Sherni. Nowadays, Sherni movie has come, so I would give you that award. Maybe you are really a Sherni. And it is our privilege to have you here, ma'am. And we all are waiting for your talk, your inspirational talk. And now I would request you to start your talk and proceed further. Thank you, ma'am. Over to you.
Thank you very much, Kavita, for your kind words. I'm really humbled. And I would like to thank the organizers, BioSetup, who has given me the opportunity to talk to you of a little research what I have done. Thank you very much. So can I start the slide share?
Yes, ma'am. You can start.
Can you see the slides?
Yes, ma'am. Perfect. Visible.
Okay. So today I will be talking to you on good bacteria to enhance the gut health. This is our CFTRI Institute in Mysuru, where I have done my research. As you know, in the plant kingdom, bacteria, algae, and fungi, these are the three which are utilized in manufacturing as well as in service industries. If we look at bacteria, there can be beneficial bacteria and there can be harmful bacteria, which can cause diseases, so many tooth decay and toxins and other things. But today we are going to concentrate on beneficial bacteria. So what are these useful microorganisms? These lots and billions we have microorganisms which have been discovered for our survival, which are good for us and also good for the industries. If you look into the nature, in the oceans, if you see, there is a blue-green algae, the cyanobacteria. These provide oxygen. Today, we are so much deficient of oxygen and we are running oxygen express, as you see in this COVID times, because oxygen deficiency is there. So these blue-green bacteria they have the ability to produce oxygen in the oceans. In nature, we have seen in soil, our Prime Minister is talking about soil card. Why is it important? Because there are bacteria in the soils which help the plants to grow. Similarly, in legumes, like we have beans and other things, where there are in the roots, if you can see here, in the roots, there are small bacteria which are called as rhizobacteria, which fixes the nitrogen and allows the crop to grow in a better way. In the food industry, if we see, and yogurt, which is a fermented product due to lactic acid bacteria. There are bread. Every day we eat bread and dough products, which are a yeast fermentation, and alcohol, which is made with the yeast Saccharomyces cerevisiae. So beneficial bacteria are probiotics, which everybody is talking today. It's a hot topic of today's nature because everybody wants to go for a natural thing. So what are these probiotics? These probiotics are live microorganisms which, if taken in adequate amounts, what do you mean by adequate amount? It should be minimum of log of 7 CFU per ml. Then they give the beneficial effect. So these can be, and they maintain the microflora in the gut. Today, if you see, the major diseases of concern are malnutrition. Our 40% of the population in India is malnutrited. Then we have obesity, amnesia, Parkinson, osteoporosis, cancer, gastrointestinal problems, aging, hypercholesterol, cell damage, cardiovascular diseases, dental caries. So can we have a one-stop answer? Yes, it is probiotics. These probiotics can maintain the microbial balance, then can increase the immunity, they can have the fitness. So what are these probiotic cultures? These are gram-positive and non-spore formers. It can be a cocci or it can be a bacilli. It can be anaerobic or it can be anaerobic. It is catalase negative and non-motile. End product can be either lactic acid, or along with lactic acid, we can have butyric acid, propionic acid, acetic acid, and things like that. So major probiotic bacteria are like bifidobacteria, streptococcus, lactococcus, lactobacillus, which we also find it in yogurt. So these probiotic properties should be that they are easily cultivable. They should adhere to our gut intestine, because if they adhere, then only they can give the beneficial effect, that. And the logarithmic phase should be longer, which means they should be viable for a long time. Only the live cells, if they go in our gut, then only they can give the beneficial effect. And there are a number of benefits. They can improve our gut microflora, they can enhance the digestion, they can improve the lactose tolerance, because so many people are not able to take milk or milk products because they do not have beta-galactosidase. They can have gut nutrients, it can reduce cholesterol, anti-tumor activity, and also it is anti-cancerous. But what is the mode of action? Mode of action is competitive mechanism, or it has antimicrobial activity where it can change, you can remove the pathogen from the gut and go and adhere to the mucosal membrane of the gut. And it can also stimulate the immune system. I have a lot of work which I have done in the laboratory, which my group, so that is what I'm going to show a number of slides, so that most of you are students, how you can go about it. And then there are functional foods. Everybody is talking about probiotic functional foods. What is a functional food? Which has a function. Say, for example, somebody wants for reducing the cholesterol, or somebody wants to decrease the triglyceride, something like that, or obesity. So a food which can have the property much higher than the nutrients which it has in the ingredients. That is a functional food. As you see in the market, today Amul has come, Danone has come, Mother Dairy has come with different products. And the functional food market is expected to reach $8,000 billion by 2025 and around $12,000 billion by 2030. It is very important that the strain, what we select, should come under GRAS category. What is a GRAS category? You know that generally recognized as safe. Because the human consumption is there, it is very important that these organisms which we select to incorporate or supplement in a food, they should be safe. So it has to go through a GRAS category. And purchase motivator, because even today, there are many people, if we are fermenting idli batter, dosa batter, or some other dal or something, we are dokla or something, we eat it easily. But when we say a bacteria is added from outside, there is a little apprehension. So that we have to remove. The attitude of people, that should be changed to see that how it can benefit our body. And the harmful bacteria, we are so much worried because we do not have good storage places for our fruits and vegetables and grains. These harmful bacteria, they spoil the food. They affect the animals and the plants. Their skin, the GIT becomes weak because of harmful. Today, what is this corona? People are telling that you should increase your immune system. Why is that? Because this coronavirus is weakening. It is a harmful virus. It's not a bacteria, but it is a virus. It is weakening our immune system. So how can we increase our immunity by a natural means is very, very important. So probiotic plays a very major role there also. And what are these harmful bacteria? Are Pseudomonas, Shigella, Salmonella, Bacillus, diphtheria, Clostridia, Staphylococcus, Vibrio cholera. I remember, there used to be a time when we had this swine flu. We had this diphtheria, but today it is not there. We had smallpox. When we were small children, we used to find many children had those smallpox also, but today it is eradicated. I'm sure in a year or so, this coronavirus also will get eradicated because a lot of people are researching on the vaccines and we are already vaccinated with the antibiotics. So as we take the food, with different types of food, we take the phenols, we eat the carbohydrates, and we take the bacteria, the good bacteria. So it goes into the gut and there is fermentation. Now what happens? Different metabolites are formed and there is a change in the microflora, which ultimately gives us the health effects. What are the desirable qualities in a probiotic culture? It's very important that it should go through a valid taxonomic system. What is a valid taxonomic system? It should go through Burgess manual that we call as a Bible of bacteriology. And it should be viable with a sufficient amount of time. Say, for example, we say that my product is good for, it has a shelf storage for 6 months. What does it mean? That the culture of the bacteria, good bacteria, we have putting it into the product is viable till 6 months. That is very, very important because I have seen many products, because in the lab we brought and one student did her MSc dissertation only in this subject. And nobody wants to build the cat, but there are little apprehensions for these things. Then it should be genetically stable. Today you see now Delta mutant has come for coronavirus, isn't it? Every day we are seeing the different type of a virus is coming because the mutation is happening. So it is very, very important that the culture which we are adding into the food should not mutate. It should be genetically stable. It's not easy that immediately a person in the laboratory, a scientist or a student, isolates a culture and immediately goes into the animal studies or human cell lines. So it is important to do some in vitro test, in vitro methods. What is that? First of all, we can simulate as per the intestine, digestive tract. Then we can have the growth characteristics of the culture and also check if it is adhering to the intestine or not. These are the work which we have done in the lab. So I'm trying to show you that how we can have a probiotic culture. If we are simulating, in stomach, the culture stays for half an hour. And the pH is 2. It's very, very low. You can imagine the harsh conditions, how a culture has to go and sustain itself, colonize with the intestinal membrane, and still multiply. Now, we have taken, say, for example, 8 CFU per ml, but after 30 minutes at pH 2 in the stomach, we have just simulated these conditions in our flask. Only this much, 7.2 worth. This we took 7.3, we took in the jejunum. Now in jejunum the pH suddenly becomes 7.5. You can see from the stomach pH, actually it can go to 1.5 also. So from low pH it is going to 7.5, and a bile salt mix, it is not one, because many people are doing experiment with Oxgal, only one. It is not like that. There is a It's a 4% very high concentration of salt, bile salt mix, which has cholic acid, trichloroac acid, so many taurocholic acid are there, and different concentration. It has a different proportion. So in that particular proportion, one should mix and then check out at 4%. And we found only this many number. It stays in jejunum for 4 hours, and we found that around 7.15 CFU per ml was remaining. So these have died. We are not taking. This we take and again simulate to the small intestine conditions where the pH goes still higher at 8, and the bile salt concentration slightly decreases to 2%. Now what we did is, this is small intestine where the culture stays for 12 hours. Now in these 12 hours, again we found that some number of cells were dying. And after 12 hours, which sustained, we took it again to the large intestine, because here the cultures start adhering to the intestine. And this is the colon, the large intestine, where the pH goes to 9. Can you believe it? From pH 2 to 7.5 to 8 and then to 9. Similarly, the bile salt concentration from 4% to 2% and 1.5%. The culture has to sustain all these conditions. And we found, and here in large intestine, it stays for 24 hours. So this culture we took as a probiotic to do further work, because this is possible to have antimicrobial activity. We also did, this is an established method to look into the adhesion property, because we cannot immediately do the rat experiment or something like that. And these are called as hydrophobic interactions. So we took different hydrocarbons at 7% and checked the OD after 2 hours to look into how was the percentage of adhesion. The culture which had good adhesion only was selected further as a probiotic culture. Now I'm telling you to have a good taxonomic as per the Burke's manual. So what it is? Look into the morphology, this gram staining, catalase, temperature, growth with sodium chloride, arginine hydrolysis, and sugar fermentation of different sugars, which ultimately, this is the scanning electron microscopy of leuconostoc. This is all the work we have done in our laboratory, and these are Lactobacillus plantarum cells. Then we also did the short chain fatty acids. As you know, the PUFA, everybody's, nowadays people are saying don't eat ghee, don't take vanaspati. Why? Because you take sunflower oil or a safflower oil, because it should have PUFA, that is polyunsaturated fatty acids or short chain fatty acids, which usually consist of hexadecanoic acid, pentadecanoic acid, oleic acid, butyl, octanol, propanol, and things like that. So what we did, we took our branded milk, which is Nandini in our Mysore city, and fermented milk with our probiotic culture, and we brought one control culture from the culture collection center. We got it, and we tried to see what are the short chain fatty acids. Of course, milk had little amount of short chain fatty acids, but our fermented milk had lots as compared to the control. So these compounds have activity against the ulcers, microbial invasion to E. coli, Salmonella, Pseudomonas, Enterococcus, Bacillus cereus, Candida. It's and Streptococcus. It's established work done by Charrington in 1991 to show that it has activity against ulcer. Then, as we get diarrhea or some other problem, you must have seen that doctor gives us antibiotics. Now, today it has become more fashionable, according to me, that doctors have started giving lactic acid bacteria capsule along with that, with antibiotic. Now, with antibiotic, when lactic acid bacteria has been given, because antibiotic, what does it do? It kills all the good and the bad bacteria in the intestine. And when there is no bacteria, then they say, you take some curd and you take khichdi or something like that. So the digestion is better. And also with curd, we get some microorganisms. But at the same time, if they are giving a probiotic capsule, it is very, very important that at certain concentration, and then doctor tells us, you take 30 mg 3 times a day or 40 mg. So it is also important to check the quantity of the antibiotic. So what we did is, and what should happen? It should be resistant to the particular antibiotic what doctor is prescribing, is it not? So the high media has got a octadisc where it has different antibiotics at different concentrations. So at different concentrations we tried different antibiotics, and the concentrations we changed, which doctors usually prescribe, and then check the inhibition. Say for example, if doctor is giving chloramphenicol and it has an inhibition zone of 32, that means this particular lactic acid bacteria will not work against the antibiotic chloramphenicol. But if doctor gives cephalothin, then this particular lactic acid bacteria will work because it is resistant to that particular antibiotic and it will not get killed. Then it is very, very important. These all work we have done in our laboratory. My students have done the work. The stability and viability of lactic acid bacteria. There are different adjuvants because, if we are adding it into a product and say, for example, we say a shelf life of 6 months, but it dies within 1 month. What do? You have already put it in the market. I've seen many products actually, I don't want to name here, that the culture lied. So we wanted to see how is it possible that we can have these lactic acid bacteria stable and viable for a life. See, here you can see we added some adjuvants. These have lysed. Where this is all SCM which we did in CFTRI, and here you can see these are all good viable cultures. So what we did is we added different adjuvants. We have published this paper, it's there in the domain, with cysteine hydrochloride, ascorbic acid, tryptone. I mean, each slide which I'm showing you is almost a 3 years work, each particular slide, and we have published papers. And casein hydrolysis. We also did with the cryoprotectants by freeze drying, because many times in the capsule they add freeze dried lactic acid bacteria. So we did it with lactose, with PEG, PET, sucrose, and we found that the initial cell concentration which we had put it after lyophilization for 3 months, it was the same CFU. However, the number slightly reduced. We tried immobilized microbial weed using sodium arginate and calcium chloride. But we found that with the adjuvants and also lyophilization was found to be the best. Then we looked into the adhesion property. Of course, this hydrocarbon I have told you. Then what we have done is we inoculated, this is the intestine of a rat, we inoculated our cultures. You can see these small, these are lactobacillus and pediococcus, different cultures. I mean, my different students use different cultures which we isolated ourselves in the laboratory, and this showed that yes, it is adhering. Then when we did the rat experiment, can you see here? These are the cultures which are adhering to the intestine of the rat. So we did after feeding, then we have dissected, and then we have seen the intestine, the SCM, where we can see that yes, the culture. So, we say yes, these are probiotic culture, and also it is important that the probiotic culture should have a S-layer protein. And what is a S-layer protein? It is with lithium chloride, and it has a kilodalton, very low kilodalton, around 30 to 40 kilodalton protein. This is the work done by Gill et al., where they found the immunity, and they found that interleukin levels and also the interferon levels became very high with different lactobacillus and bifidobacterium cultures. Then let us look into the dental caries. Now, what happens in dental caries? It's an infection by Streptococcus mutans. And what happens, because when we eat and we do not brush in the night, or maybe during the daytime also, there is a lot of sucrose, and which produces lactic acid, and these lactic acid bacteria, they feed on it, and then lactic acid is produced, and then the blood starts coming. There is a plaque. So what it can happen is these probiotic cultures nowadays in Germany and many other European countries, I have seen when I had gone last, that their toothpaste they have with a probiotic toothpaste. So what happens, especially for the small children, they tell them to brush in the morning and before going to sleep, because they eat a lot of nutrient bars and choco bars and things like that, where there is a lot of starch on the teeth. So if they brush with the probiotic toothpaste, the probiotic cultures go and stay there, and this pathogen, which is called as Streptococcus mutans, it comes out. So the organisms usually in the human host are homofermentative, which produce only lactic acid bacteria, and heterofermentative, which produce other acids along with lactic acid bacteria. Now we call antibiotics when we talk about synthetic, which can kill the pathogen. Similarly, when we have antimicrobial metabolites by probiotic cultures or lactic acid bacteria, we call them as bacteriocins. Say, for example, Lactobacillus acidophilus is called as acidocin. Gasseri is called as gassaricin, and reuteri is called as reuterasin. Then we also looked into the antimicrobial activity. When we are talking about the bacteriocins, we wanted to see which particular bacteriocin can it be there. Because I had a number of projects from DST, the national projects, and as well as international projects, where we isolated these bacteriocins. So we looked into with different pathogenic strains like E. coli, all common pathogens, and we looked into the different inhibition zones. These are all the work is done in our laboratory by my students, and we found the different zones. Any zone above 3 mm is known as positive. And we also had a control sample from the dairy, and then we compared it with our cultures. Similarly, around in 2009, there was a news in Times of India that a deadly strain of cholera had hit India. At that time, I had written a project how we can have a bacteriocin against cholera, and I had got a bilateral program from DST with Argentina, where we tested our lactic acid bacteria and also Bacillus subtilis. And this, you can see that, how the Vibrio cholera is getting on mucin. We had tried to see how it can come on a fluorescence microscope. And when we added Vibrio along with lactic acid bacteria and Bacillus subtilis, we have a patent on this, how, you can see the number of cells have got reduced. Then we had another project where we, from Shigella, we isolated, against Shigella, we isolated a bacteriocin from pediocin, and we had different methods of ammonium sulfate precipitation, but we found that defatting with chloroform methanol was very good, and we got different bands. Similarly, then that we anti-Shigella fraction, we did STS page, and we found that the molecular weight was around 22,000 kilodalton. After that, we took the fraction, and this HPLC, we again collected different fractions, and for each fraction, we did anti-Shigella test, and we found that a peak at 9.5 had anti-Shigella activity, which we confirmed again with LCMS, and we found that yes, this is the active fraction which can be called as a bacteriocin. So this particular fraction had the activity. Similarly, we had done with cholera also. So coming back to the literature, if you see about the anti-cancer effect, we say people who are eating a lot of vanaspati or high protein diet, very high protein diet, they usually get cancer. And why does it happen? Because this high animal fat and the protein diet produces these enzymes. These are pre-carcinogens in the intestine, like glycosidase, beta-glucuronidase, azoreductase, nitroreductase. Now what happens? The pre-carcinogens, they get converted in the colon and get converted into carcinogens. But if a person is taking probiotic culture, it can be prevented to a large extent because it does not allow these pre-carcinogens to get converted into active carcinogens. Similarly, for the peptic ulcers, it was very difficult actually to get Helicobacter pylori, and it was not allowed in the lab. Actually, I tried this, but this is a work of somebody else in US where they found this Helicobacter pylori, which is a pathogen and can cause peptic ulcers, can be cured, can be prevented rather with probiotics, because antibiotics have a very negative effect and a lot of side effects also. Similarly, antibiotic associated diarrhea. There is an imbalance as doctor prescribes us antibiotics, without, say, for example, probiotic, or without B capsule. Otherwise, the earlier doctor was prescribing us B complex capsule, if you remember. So what happens, it is trying to balance the colonic microbiota. But however, if there is a diarrhea, there is an imbalance in the colon microbiota, and this causes a lot of water. Water doesn't get retained. So probiotic is possible to prevent this AAD. Similarly, allergy. Allergy, if you see, it makes a endogenous barrier and to alleviate the intestinal inflammation. It also reduces the Th2 cytokine response. Then we also did a work, this also we had published on beta-galactosidase. Even, I have seen in my very near relatives and all, so many of them, they are not able to eat milk products. Why does it happen? Because they do not have beta-galactosidase enzyme. And as you know, milk has almost 70% of fructose. It's not able to digest it. It's not able to break down. But if galactosidase is there, which can be provided with a probiotic culture. We did a lot of work, it's almost a 3 years work, and we could find that yes, it was able to do good results in mice. This is a work of Jacobson, which I just want to show you that, see, he has taken some 44 strains, out of which he tried only at 2.5 pH for 4 hours, and only 29 strains they stayed. And similarly for bile salt stability, only 16 strains, where he has tried only Oxagal at 0.3%, where I was telling you 4% bile salt mix, and adhesion to cacocells at 4 only out of 44. Similarly, the serum cholesterol, this we found that in the whole milk, yogurt prepared with a whole milk and yogurt prepared with a skimmed milk. If we are talking about the serum cholesterol level, you will find that in skimmed milk with yogurt, it's very, very less. So that probiotic cultures can definitely prevent the serum cholesterol. Similarly, advantages to ruminants. The cattle, every time they are eating these plants, so it can inhibit. If probiotics are given, in fact, we had a project with Suguna Poultry Farms also, where we made a feed for the chicken to give them more eggs. But in ruminants, I'm trying to tell you that it can inhibit the methanogenesis because it produces a lot of methane gas. There is a gain in weight. Milk production becomes high. Genetic manipulation of the rumen bacteria does not happen, and it can degrade the phytotoxins. So we did to see the effect in animals before we come to a conclusion. And with the diarrhea, we induced the diarrhea with lopamide, and then we had different concentrations of a probiotic culture where we found that the fecal moisture reduced and also the body weight increased. See, this is the diarrhea culture in our cage, and this is the probiotic which I had to hold the tail, and I told my student to take the photograph. If you can see, it's so active and so healthy. We also looked into the spleen, heart, and lungs, where we found there was no significant deviation, telling us that yes, that it was normal. And also we looked into the diarrhea animals for creatinine, alkaline phosphatase, and HGOT, where we found that there was no liver damage and no kidney damage. So there was no damage to the organs also. Then one of my students, she had prepared probiotic Ragi drink to reduce the cholesterol and the triglyceride, where we won the best paper award in Malaysia. And here we found that the cholesterol levels and the triglyceride levels, they reduced quite a lot with probiotic Ragi drink. Actually, Ragi is very much common in southern India, southern part of India. And we also prepared one fermented food, probiotic food, where we added Ragi and wheat, because in northern part of the country, they use a lot of wheat, whereas in southern part, they use Ragi also. So, and we found the mixture of Ragi, that slide I have not put it here today, Ragi and wheat is also very good. And we made it sort of a halwa sort of a thing, which can be given to all age of people. So this we found that the product had number of antibacterial compounds when we looked into the therapeutic importance, which had bactericidal activity, like decanol, we got antibacterial activity, octanol, we got anti-inflammatory activity, hexadecanoic acid, which were in the probiotic product and not found in the control sample. Now, all of you are aware of HIV. Now, how does HIV happen? It is an autoimmune deficiency, which happens because of many pathogens together, like Clostridium, Shigella, Campylobacter, Salmonella, Staphylococcus, Mycobacterium. Now, what this probiotic does, if the probiotic is taken to the HIV patients, what happens? It forms a layer on it, an integral layer, where it does not allow these pathogens to come, and the CD4 cells do not die, and the immunity stays back. It helps in Th17 cells, a good GALT. What is GALT? A good gut-associated lymphoid tissue. Otherwise, this lymphoid tissue, also there is a loss. So it is a multi-purpose tool which can be a key component to prevent even the sexual and the reproductive health. Many probiotic foods are available in the market, especially by Stonyfield. I'm talking to you about all over the world and also in India. So this majorly, it has Streptococcus thermophilus, Lactobacillus acidophilus, and Lactobacillus bulgaricus. You know, many of you may think, what is the difference between a yogurt and a curd? When a yogurt is made, it has only two cultures, usually the Streptococcus thermophilus and Lactobacillus bulgaricus. But when we prepare curd in the house, we have a previous day's inoculum, which we just add, which has a multi-fold antibiotic, multi-fold lactic acid bacteria. Another they have made is a low-fat chocolate yogurt, fruit flavored with goat yogurt and in the squeeze tubes. Lalus have come out with a yogurt milk. In fact, we have a paper with my student Gayatri where we worked on goat milk probiotic food, and we used to get, my student used to get that goat milk from Dubai. It was so difficult, and it used to come by flight to Bangalore, and from Bangalore again we have to come to Mysore. So we used to take ice buckets, you know, ice thermocol things, and by the time, many times when we come to Mysore, what we found, it used to get curdled. The goat milk gets curdled very fast. So one has to, but it has so many nutrients that, you know, it is a total food and it can increase the immunity to a large extent. Goodbelly has come out with organic fruit juice. Again, for if somebody is working with fruit juices, you have to be very, very careful because the pH goes down quite a lot. And there they have added Lactobacillus plantarum. It's in different flavors. Similarly, Attune have come out with chocolate and granola bars. They are called as wellness bars because they increase the immune system. And Indian Kashi Vibe is a wellness cereal. It's not only the milk products, but today people are talking about cereal foods, like I told you, ragi and wheat and things like that. Then Danone, as you know, it came in 2004 in India, in Haryana, and it's a dairy beverage. It's available in all big malls, and it contains Lactobacillus casei shirota cells. Then we have a, you know, in southern India, if you say we eat curd rice, masru anna, it is called as. It's very, very healthy, but many people around the nation and around the world are not much aware. But as you can see, the Ricera, they have come out with Ricera rice yogurt, where they add rice and yogurt, which contains Lactobacillus bulgaricus, Streptococcus thermophilus, Lactobacillus acidophilus, and Bifidobacteria bifida, which is very, very nutritious and increases the immunity. The dosage, as I told you, can be in a capsule form, it can be in a stick form, it can be powder, or it can be chewable tablet. It can also be individualized, I was telling you, functional food, or customized in the form of any way a person or a patient can take it. Then we talk about multi-probiotics, where, you know, there are group, because if you are taking a single probiotic, individual probiotic, or if you are talking about multi-probiotics, which means, you know, different microorganisms into one, which means, you know, you need to know the one probiotic should not cut the other probiotic, right? So it is very, very important to study the growth, to look into each phase, growth phase, because each microorganism, each probiotic culture should be in the logarithmic phase. So if you are looking for a multi-probiotic, it is very, very important to see the different multi-biota and look into the different things. Then only you can have a symbiotic association. Now people may think, do I have any side effect of this probiotic if I take? Yes, of course. If the immunity is very low, if the age is very high, sometimes it can happen, because there are, today also for COVID vaccine, people are talking if there are comorbidities, then person should not take. So it is very, very important to look that the person, if is having something or taking some drugs, should not take. If the age and they have some other diseases, they have to check out with the doctor and do not take just like that. Compromised immune systems can have rash or fever or bloody stools. I am not discussing on prebiotic, but just for your information, I made some 2 slides on prebiotics to tell you that prebiotics are non-digestible components which can stimulate the growth of probiotics in our intestine, in our gut, which is usually the garlic or the onions or the whole wheat or rye or barley. What does it do? It stimulates the probiotic culture. And the common probiotic culture which we have been taking from time immemorial are milk, soya, tofu, sour cream, buttermilk. I remember, buttercream in southern India, we have majjike, which is very good. And after having a heavy meal, they always take curd rice, and after that they take majjike. I'm sure, I think if you really look at it, southern India food is very, very nutritious. And the idlis and the dosas, then doklas in Gujarat, and vadas and curry in North India are all containing probiotic cultures. In India, the probiotic research is associated. It's happening in CFTRI, where I work, National Dairy Research Institute, Karnal, Haryana, Institute of Microbial Technology, Chandigarh, where we have deposited all our cultures, National Dairy Development Board, Anand in Gujarat, and Nestle Private, Panipat, Haryana. So why should I take this probiotic culture when I'm taking antibiotics, alcohol, and a lot of stress, It destroys the gut microflora. The emergence of antibiotic resistance. All this is a one-stop solution is probiotic. Kajol has been very active in featuring on TV and radio to talk about Yakult, and also by Mother Dairy, Be Active Probiotic. This, I want to show you some slides where are different age groups, if they don't have any other problem, these probiotic, a lot of people have worked with human clinical trials, and we have seen from 2 weeks, can you see here, to 86, and also the 94-year-old person with different dosages and different cultures which have been found successful. So, to get a probiotic food, one needs to have a strain identification according to Burgess manual, deposited in an international culture collection center. Phase 1, have an in vitro analysis and characterization, functional characterization. If it has PUFA and things like that, then have animal studies. Phase 2 with a double-blind, placebo-controlled studies, and then 3rd phase is effectiveness, to prove it should be appropriate with a standard treatment of a specific condition. A probiotic food should have a proper labeling of the genus, of the species, of the strain, and its designation. It should have the minimum number of viable cells at the end of shelf life, which I'm not finding too much. It should have proper storage conditions, at what temperature it should be kept, which are not having in many of them, and the corporate contact details if a consumer has a problem. So it is critical to forge not only the scientists, but it should include the scientists, the advocates, the funding agencies, the government agencies, the regulators, the healthcare providers, and the community, all of you, each citizen of our nation. So the recommendation of a probiotic culture is a well-defined culture. It should be safe for human consumption for different target groups and should be persistent in the GI tract with antagonistic activity against harmful microorganisms. It should be technically feasible and stable to have on a large scale and with documented stain characteristics. So I'm sure the probiotic revolution has begun to have a habit which can really benefit the overall health by taking probiotics. This is my interview on the TV on Doordarshan Science with Probiotics Friendly Culture, which came on Doordarshan, and this is my interview at Red FM Radio. These are the books, technical books, which I have authored and published by NEPA. This probiotics and the role in improving human health, making food safe and free from pathogens, utilization of agricultural waste for the production of high-value food products, traditional foods, and nutritional security. So I thank you all, and let us live well with probiotics. Thank you very much.
Ma'am, there are a few questions in the chat box.
Yeah, sure.
Should I read it?
Now, one thing, what should I do? I want to close my slides and come back so that I can talk to the people.
Okay. Ma'am, on your screen there, just the 3 dots, click on that and do stop sharing.
Where is it? Stop sharing. I'm not seeing.
There is, ma'am, there will be 3 options: mic, camera, screen. So click on the screen and stop that.
Sorry. More options I got. No. Stop sharing. Can you stop it for me? No.
Yeah,
Okay.
I'll just check.
Right.
Yes, ma'am. Right.
Can I come to the big screen? Probably, I thought. Yeah. Can I, one second. Can I enlarge it? More options I'm seeing.
Ma'am, there will be 3 icons wherein you are sharing your video, your audio, and the last option is your screen sharing. You need to click on that and stop sharing. You have to do.
That only I'm not able to see. Yeah, now maybe.
Yeah, but you need to cancel it so that your screen, yeah, you can click on
One second. One second.
Yeah.
1 second. No. It's all right. It's okay. These slides have gone, but
ma'am, you can see on this taskbar down, you can see that PPT presentation. You cancel it, it will go.
That I have done. Now I want to make this full screen. It's okay. Let them ask the question. It's all right. Otherwise, we are wasting our time. It's okay.
Yeah.
Hello.
Yes, please.
Yeah. Good morning, ma'am. It's very nice and very informative talk. We are very happy attending in this webinar. Ma'am, this probiotics also nowadays by the using of multiple antibiotics, the probiotic, a few commensal bacteria become a multiple antibiotic resistant. So this probiotics also in future maybe gets in a antibiotic resistant.
Yeah, I told you, no, I showed you one slide where we have worked with different antibiotics, and it may be resistant to some and it can be sensitive to some, you know.
Ma'am. Yes, ma'am.
So it can be resistant.
So,
One has to, that is why I'm telling, when the doctor gives, we have to first get it tested, right?
Yes, ma'am.
Yeah, the next question, please.
Dear participants, do not unmute yourself. I'll be taking your questions and ma'am will be replying for the same. Ma'am, the question is, can you please elaborate on probiotic in fermented rice? Can everyone eat it?
Fermented rice.
Yes,
Hello.
probiotic in fermented rice.
Yeah, we are eating every day, no, in dosas. That is known, is it not? Fermented rice,
Yes.
everybody. Yeah.
Okay, the next question. Ma'am, how does the
Excuse me, ma'am.
microbial cell?
Excuse me, ma'am. I just asked the question. It is not about the fermented rice. It is like the old rice, the previously they cooked rice that we store it
Okay.
in the water and next morning we eat it. So many say that people with the diabetics cannot eat that, but it is full of probiotics. So what is your opinion on it, ma'am?
Okay, okay, that way you mean. Okay, I think rice has a lot of carbohydrates, So that way one has to check for the diabetics. Otherwise, it is good. But diabetics, because no sugars, they should not take. Depending on the case or the chronicity of the patient, you have to ask the doctor. Because rice has a lot of starch.
Thank you, ma'am.
You're welcome. Yes, any other question?
Yes, ma'am. Just a minute. The next question is, ma'am, can you please elaborate? Is the probiotic working against colon cancer?
Of course, it can be prevented. Definitely. I showed you one slide, right? A lot of work is going on that.
Okay.
Yeah.
The next question is, ma'am, how does the microbial cell remains viable in probiotic drinks such as
Yes.
Yakult?
Very good. Very good
What
question.
preservatives can be used that does
No,
not
no,
affect the viability?
no, no. I told you not to use the preservatives, but as I told you, I had shown you the slide of our work in the laboratory where you can add some adjuvants. Now, what are these adjuvants? They are like antioxidants. Everything, they are telling, the cell should not die, you should have some antioxidants, right? So you can add like cysteine hydrochloride, ascorbic acid, something like that, which can enhance and also study the growth phases. So if the logarithmic phase is more of the culture, then it will be viable for a longer time. Okay.
Okay, thank you, ma'am. The next question is, can we safely say that probiotics are becoming the effective alternative for antibiotics?
Yes, definitely.
Okay.
Definitely.
Which food have rich probiotics?
Sorry?
Which food have rich probiotics?
No, that you have to see the label. I can't tell.
Okay. Okay. Ma'am, can probiotics cure liver diseases?
See, there is nothing like cure. It's prevention, right? Nobody can guarantee cure. That way, even antibiotics, they cannot say cure. You know the difference between cure and prevention? So it's only prevention. Yeah.
Okay. Ma'am, can we use probiotics into regenerative medicines? And please explain how probiotic bacteria being used in cancer prevention.
Any form they can take, no? They can take it either in a powder form, they can take in a functional food, if it is there available in the market, or they can take it the way they want. They can add it in a yogurt, they can take it anyway. But it should be written in the
Ma'am,
label. Yes, it is for the anti-ulcer or anti-cancerous or whatever it is.
one question from Dr. Raja Gopal. He's
Okay.
asking, what is the difference between prebiotic and probiotic?
Okay, I had shown you one slide, doctor. See, these prebiotics are, these are mainly oligosaccharides, like FOS, something like that, fructooligosaccharides. And these are only helping the probiotics to grow more. And they do not have, they just like fiber. Like they say, for good gut, this thing you should take isabgol. I think you must have seen on the TV, say take isabgol. Even that actor comes and he says you take isabgol. They're all fibers which are available in different cereals or onions or say barley or say garlic. Yes.
Okay. Ma'am, the next question is, ma'am,
I
does
think all these
probiotics
questions,
have...
excuse me, all these questions I have already explained in the slides. I think the same questions are being asked. So kindly you can provide them the slides, and if there are any extra questions out of the slide, then they can mail me. Okay.
Okay,
All these
sure, ma'am.
questions, what I'm answering is already I have told in the slides.
Okay. One minute. Dear participants, you can add your questions in the feedback form, or you can send the questions in the WhatsApp group and I will mail it to ma'am. So all your queries will be solved. Okay.
So thank you very
Thank
much.
you, ma'am. Thank you, ma'am, for sharing your deep and insight knowledge about the topic. Now I request our founder coordinator, Argyadeep Das sir, to enlighten our participants with his golden words. Over to you, sir.
Okay, thank you. So, am I audible?
Yes.
Yes, sir.
Okay,
You're
thank you.
audible.
So, a very good morning to one and all. My heartfelt thanks to our guest speaker, Dr. Renu Agrawal, ma'am. So, I thank you for accepting my invitation for the webinar. It was an academic fest. The topic of your research is so relevant and useful in this time. So, and the way you shared your knowledge regarding the topic was so impressive and encouraging. Thank you for making the time to share your work with us, and a big thank you for answering the questions of all the body researchers and students. So it's give me an immense pleasure to deliver vote of thanks to today's speaker, Dr. Renu ma'am, and also all the participants. Once again, I thank you, ma'am, for giving such an inspirational talk. Your dedication and work in the field of not only science but also for the betterment of farmers and society is really commendable. Hats off to you, ma'am. I look forward to your guidance in future too. And also thank my organizers, event coordinators, Dr. Ritu Chandel, ma'am, Dr. Kavita Pal, ma'am, Dr. Arpana Sharma, ma'am, and our organizer, Miss Neha Mahajan, Miss Ananya Chakrabarti, Miss Shreyans, and our faculty member, Miss Vinita Tiwari, Miss Somuju Bhargavi, Miss Priyanka, and our invited guest, Dr. Amut, and my dear students and researchers, thank you for joining and invest your precious time for listening the talk and support us. Last but not least, and on the behalf of BIOSETUP Life Science, I thank each and every participants for contribution. This webinar is a successful one, and I look forward for arranging many such events to let people know how progress made in science. Thank you, everyone. Have a good day. Thank you, ma'am, once again.
Thank you. Thank you very much. Kindly send me the recording and okay.
Yes, ma'am. I will provide you after this session.
Fine. Thank you. I'm leaving the meeting now.
Okay.
Yeah, sure, ma'am. Thank you.
Thank you so much. Thank you so much, ma'am.
Thank you.
And thank you all the participants for joining with us. I'll be sharing the feedback link. A humble request to everyone to please fill the feedback form and then exit the session.